Let's start for the background of PM2.5. According to the World Health Organization in 2021, 6.7 million premature deaths occurs annually as a result of air pollution, both indoors and outdoors. A key indicator of air pollution is PM2.5. What is PM2.5? PM2.5 is an atmospheric particulate matter that is 2.5 micrometers or less in diameter. And PM2.5 induces inflammation, which can lead to lung cancer. And next, for the background of molecular docking. Molecular docking is a computer-based program that predicts the binding affinities of a protein-ligand complex. Why we use molecular docking? We use molecular docking because it simply saves time. Instead of testing 1,000 ligands in the wet lab, we could just use molecular docking to predict the best binding ligands and test out those ligands in the wet lab afterwards. And now for the background of interleukin 1. Interleukin 1 composes of interleukin 1 alpha, interleukin 1 beta, and interleukin 1 receptor antagonists. However, interleukin 1 receptor antagonist does not emit an inflammatory response. So that is why, in our research, we'll only be focusing on interleukin 1 alpha and interleukin 1 beta, which are cytokines with similar pro-inflammatory properties because they both bind to the same receptor. Interleukin 1 alpha is continually expressed in the human body. However, interleukin 1 beta is only expressed when in response to inflammation. And studies have also shown that the inhibition of interleukin 1 have been shown to reduce cancer metastasis. So here's a PM2.5 induced inflammation cycle so you can clearly understand. So firstly, when PM2.5 enters your respiratory tract, it damages the lining of your lungs, which causes the polarization of M1 macrophages. The polarization of M1 macrophages causes the expression of interleukin 1 beta, which then causes inflammation. And the inflammation damages cells. And now the dead or damaged cells releases interleukin 1 alpha. And interleukin 1 alpha acts as an alarmin and signals immune cells to the site of the injury, which also influences interleukin 1 beta production. So that means if you're frequently exposed to PM2.5, this can create a positive cycle, which could in turn lead to chronic inflammation and turn into a tumor microenvironment. The objectives for my research is to find compounds that could bind to a protein's critical residue on interleukin 1 and act as a potential inhibitor that could reduce chronic inflammation that could lead to lung cancer. And now for the materials under the topic of proteins. The proteins that we use were sourced from the Protein Data Bank, also known as the PDB. So this is the 3D structure overview, 3D structure of the interleukin 1 alpha. The code name is 2KKI, and this is the 3D structure used for interleukin 1 beta. The code name is 1I1B. And now for the ligands. So a total of 1,584 ligands were used in this research. These pictures are just some example ligands. And the compound sets were sourced from the National Cancer Institute. And the selection of these compound sets were based off of its pharmacophoric centers, which means the ligand structure that gives it its biological activity and also allows it to bind to its receptor. And next is methodology, under the topic of active sites of interleukin 1. So the active site that we chose is site A, because site A is a primary binding site, and it is also present on both interleukin 1 alpha and interleukin 1 beta. So the parts highlighted in red
เดี๋ยวน้องช่วย
is
เพื่อน
the
เลื่อนส
active
ไลด์ก็ได้
site, also known as site A, and the yellow parts is just the remainder of the protein structure. And now for the molecular docking process. So for the actual molecular docking process, we use the program GOLD. And for the protein visualization, we use the software Discovery Studio. And two scoring functions were used in the molecular docking process, which are gold score and ChemPLP. Why we use two scoring functions? We use two scoring functions because different scoring functions calculates the scores on different aspects of the binding affinities. So this is the protein structure before molecular docking. This is the ligand before molecular docking. And in the molecular docking process, we'll be combining these two and checking its binding affinities. So this is what it looks like after. These two pictures are identical, but the one on your right, the active sites are highlighted in red, so you can see that the ligand binds to the active site. And the one on your left, it's just a regular protein ligand complex, so you can compare. And now for the results. For the results, I'll be showing you a 3D structure overview of the protein and ligand, so you can visualize what happens. And I'll also be showing you the 2D structure overview, so you can see the interactions that goes on. So this is the best ranking ligand for interleukin alpha using gold score as a scoring function. So as you can see, there are conventional hydrogen bonds, pi alkyl interactions, and also carbon-hydrogen bonds around the protein's critical residue. And next is the best ranking ligand for interleukin alpha using ChemPLP as a scoring function. So with this one, there are pi alkyl interactions and also carbon-hydrogen bonds. And next, for interleukin beta best ranking ligand using gold score as a scoring function. So with this one, there are pi lone pair interactions, pi alkyl interactions, carbon-hydrogen bonds, and also conventional hydrogen bonds. And lastly, for interleukin beta best ranking ligand using ChemPLP as a scoring function. So as you can see, there are pi alkyl interactions and also conventional hydrogen bonds around the protein's critical residue. And now for the discussion. The results show that the tested anti-cancer compounds could be potential candidates to inhibit interleukin-1, with top ligands binding to the protein's critical residue. So our findings are very valuable because currently there are only 3 inhibitors for interleukin-1 commercially available worldwide, which are anakinra, canakinumab, and rilonacept. So there's an obvious research gap and high demand for treatments against inflammation-related diseases. And lastly, for the conclusion. In summary, the anti-cancer ligand showed promising interactions with different compounds having a better affinity for interleukin alpha and others for interleukin beta. However, further lab validation are needed to confirm these compounds' practical effectiveness. These are my references. Thank you for listening, and giving me the opportunity to present here at STT50. I'm happy to answer all of your questions with the help from my co-researcher and sister, Andrew Wanasan. Thank you very much for letting me person call. So, do you have any